Hello students, and welcome to Lab 4. We're going to be talking about pH and buffers. First of all, let's go over the Lab 3 and your lab report. So, I know that this lab report is going to be the first major grade for your lab. Assess your overall lab grade. So lab 3 is the overall lab for the lab report. Okay. And lab 3 starts on page 20 of your lab manual. The lab report starts on page 30. Okay. Lab report materials, everything starts on page 30, and then the grading rubric that I use starts on page 32. Okay. So all of that will start there. All right. Keep in mind though that I'm going to add some points there for your group. All right. Some amount of points, including, so down there at the bottom where it says 40 for total lab report, I'm going to add about 10 points for that for your group. Okay, so total for the lab practical, that's going to be different. And then total for the lab report, that's going to be about 50 points. Okay, and then that all is going to be separate, apart from each other. But follow this guideline, and then you shouldn't have too much of an issue. All right? Make sure that you're following, differences between the onion juice, the potato juice, what the next experiment will be, follow all of those questions. Okay, other than that, what I want you to do is make sure that you're doing either the Benedict's test, the iodine test, or the Buret's test. Okay? So you can do either of those 3 tests, right? You're not going to do the lipids test because that doesn't have anything to do with the starch or the onion. Right? So you can do the Benedict's, the iodine, or the Buret's. Right? And all of those results should be in the PowerPoints that I sent out. Okay? And you should be able to pay attention to those. Also, what I'm going to do is, as soon as I've graded most of or all of the lab assignments and everything, I'm going to go ahead and send out some of the answers to some of these so that you can study for the lab practicals. Okay? And that's just going to be in the form of PDF document. Probably I'm going to go through and write these down and probably either scan them or do something. Okay? All right, if you have any more questions with the report, once again, try to make sure that you fill it out as a group. It's the same group that you've had, for your post-labs and everything. So fill it out as a group, and if you have any problems with your group, please let me know. There is going to be a group evaluation at the end. So you're going to divvy out the amount of points for your groups. So once you turn that in, you'll fill out the group evaluation, and then you'll give that to me. The group evaluation itself isn't for a grade, but when you send that to me, that will let me know what the grade I need to give for the report is. So, any other questions for the group or the lab report, please let me know, and I'll try to get them to them when I can. Okay, as for next, let's go ahead and get into Lab 4. All right, Lab 4, pH and buffers. We kind of talked about this a little bit during lecture. The pH scale is a logarithmic scale based on the number of hydrogen atoms, hydrogen ions in a solution. And this ranges from about 0 to 14. It can go higher or lower. A neutral scale is about 7, 6.9. Any lower than that is acidic. 7.1. Any higher than that is basic. Usually we remain internally we may remain more of a basic scale, but anything lower than that is tends to get a little bit discomforting. So we do need buffers. So antacids tend to have a buffering capacity. So a buffer is a substance that resists change in pH. So our bloodstream does contain buffers that prevents those swings out of that range. And we talked about one during lecture that you can go back and try to see what that was. So in lab we have these pH strips, right? So they're going to change their color based on the solution that you dip them in. So increasing acidity changes from yellow to red-ish, and then increasing alkalinity changes from yellow to bluish. And neutral is about green, but if it's dry, it's yellow. So if you put it into pure water, it'll turn green. So there's common substances that you can use to kind of compare pH scale. Bleaches are fairly basic. You're gonna have oven cleaners, things like that can really get into hard, dry grease and everything. So those are anywhere from 13 to 15 pH. And these contain a lot of hydroxide ions. Ammonia solution, things like Windex, these are like 10 to 11. Baking soda can range from 9 to 10. Seawater is about 8. It does have a lot of ions in it. Neutral blood is about 7.4. It can go down a little bit. Milk, urine, saliva, that goes down in acidity because you expel, in urine you expel acid to keep your urine at a basic level. Black coffee is about 5. It is more acidic. It does eat away at your teeth. Grapefruit juice, tomato juice, acidy, citric acid. Lemon juice, definitely acidic. Vinegar, acetic acid. Vinegar is acetic acid. And then battery acid, things like that can range definitely in the 0.0 scales. Hydrochloric acid, muriatic acid is another one. Things that you put in your pool to scrape away at algae. Things that you need to offset with water, distill with water, try and make it more neutral. So we use those litmus papers to try and figure out what the acidity, what the alkalinity of these solutions are. So hydrochloric acid is an acid, which means that if we tested it, the pH strip will turn red, right? Hydrochloric acid has a pH of 3. However, if we didn't know that it was what the pH was, if we weren't able to test it, we would know that it's an acid because it does have that H in the formula, right? It has that H in there. It has the readily available hydrogen that can come off that chloride, right? If this was a base, you would see that OH able to come off, right? Just like sodium hydroxide. Sodium hydroxide is a base. We would know this because the pH would turn dark blue, right? So the pH of sodium hydroxide is 14. And we can test that with the litmus paper. So when you drop the litmus paper and it turns blue, but if we didn't know that, we could see the formula, sodium hydroxide, it has that OH at the end, right? So when it comes off with an OH so that we know it's a base. So in the tests in your lab notebook, we use bromocresol purple. And bromocresol purple is a pH indicator. It's like the litmus paper, but it's a solution. Okay? So bromocresol purple is a pH indicator that turns yellow if a pH becomes acidic. Anything less than 5.2. Okay? So it's not overly acidic, so if it's 6.9, it's not going to be, that's still in the neutral zone. Okay, so 5.2 is acidic. So over here on the left, you'll see that it's turned yellow because it's acidic. But over here on the right, it's purple because in that solution there's a beaker, or there's a buffer present, right? So it's preventing it from turning yellow. It's preventing it from turning acidic. This chart right here will help you answer page 43. So buffers and bromocresol purple. So the buffer will help maintain the constant level of pH when acid is added, right? So on page 43, this is what you're going to find. Okay, and you can copy this to that chart. Right? You can answer those questions right there. So we can talk about this. The buffer will remain the same, right? It's just remaining the same acidity throughout. You see that? Even though more hydrochloric acid is added, the pH is remaining. That's all the buffer is doing. It's just remaining the same. It's just keeping the same pH. Now, if you have pure water in there without the buffer, it's dropping in pH. Because you're adding more HCl, you're adding more hydrochloric acid. So the buffer is there to keep the acidity down. And that's exactly what you do with antacids. So when you take an antacid, it's a buffer to make sure that no more HCl, no more hydrochloric acid is added to your system. Or no more acid, is added to your system and makes that acid build up in your system. These are more review questions for Lab Practical 1. So some of these are the pre-lab questions, some of these are the post-lab questions. Make sure that you review these before the lab practical. Okay. Once again, these aren't for grade, but these are just review questions. Okay. Make sure that you go through the post-lab review. And for your lab assignment, I think that's basically going to be it, is page 43. Because most of the stuff you would have had to do it during the lab. And I can post that online. If I come up with anything else, I'll post it online. But I think that's going to be it. This one's kind of a wet lab, so if there was anything else that I had you do, it would have been during lab, and we can't really do that. Okay, thank you very much. If you have any questions, let me know. All right, bye-bye.